The method shown in this video utilizes fate maps of the cleavage stage Xenopus embryo to identify single blastomeres culture in isolation to assess whether embryonic cell fate is determined by intrinsic or extrinsic instructions. First, two cell embryos are collected in which the first cleavage furrow bisects the lightly pigmented area of the animal hemisphere. These embryos are cultured. When the embryos reach 16 to 32 cells, the vitellin membrane is then removed, and neighboring cells are dissected away from the cell chosen for explant. The dissected blastomere is then transferred to a depression in an agar-coated culture well and cultured. Once the explant reaches the desired stage, it is harvested to assess gene or protein expression using in situ hybridization, qPCR, or immunohistochemistry.
The major advantage of this technique over others such as culturing large groups of embryonic cells or organ enlagen is that single embryonic blastomeres, whose developmental fate is known from lineage tracing studies, can be cultured without added growth factors or the presence of neighboring cells. Thus, one can directly test whether factors intrinsic to the cell instruct that blastomere to become the precursor of a specific tissue, or whether it requires signals from surrounding cells.
Visual demonstration of this method is critical, as the dissection of blastomeres and their transfer to culture wells requires delicate manipulations under the stereo microscope. In this video, I will be using this method to demonstrate whether maternally derived mRNAs bias cells towards a neural fate.